So in this part, we are going to discuss types of genes, and this under the umbrella of cancer causing or cancer related genes. There are two categories here, the tumor suppressor genes and oncogenes. So tumor suppressors or tumor suppressor genes are genes that help control and regulate the cell growth. Okay, so they regulate and help the cell growth and division. When they are functioning correctly, they can certainly prevent the uncontrolled growth of cells. By now we understand that uncontrolled growth of cells is what characterizes cancer. So they are doing that very important role. And a classic example of a tumor suppressor gene is certainly TP5, which is tumor suppressor, which is tumor gene 2p53. Okay, TP53 gene is a tumor protein p53. Okay, so TP stands for tumor protein 53. This is frequently mutated in a variety of human cancers. And one of the things when we have discussion about tumor suppressor genes is for that specific gene to lose its function and contribute to the cancer development or uncontrolled growth of cells, typically both alleles, both copies of that gene need to be inactivated or mutated. Okay, so if we have only kind of just one knockout in this case, or first knockout, which is, you know, one mutation in one copy, this might not cause an issue if the other allele is still functional. But the second mutation is going to lead to the loss of function in this case. In contrast, when we're looking at oncogenes, in contrast, the oncogenes are genes that have the potential to cause cancer. Okay, so tumor suppressor gene, they are responsible for the cell growth and division and so on and so forth. Oncogenes, something goes wrong, when they are mutated or expressed at higher levels, they can promote what? More cell division and growth, leading to the tumor formation. And one example of these genes is MYC gene. Now, something that makes them distinctive, I mean, the oncogenes, is that when there is a single mutation or overexpressed copy, that is sufficient to contribute to the cancer as compared to the tumor suppressors, where both alleles there should be subject to mutations. So in other words, even if one allele of an oncogene is normal, the mutated or overexpressed allele can still drive cancer development. So the protein 53 gene, and this is usually referred to as gatekeeper of the genome. The gatekeeper of the genome, okay? And so is indeed playing a crucial role in maintaining the stability of the cell's genetic material. As you see here, it is involved in many different pathways. It is involved in many different pathways. In related to cellular processes and responses. For example, the DNA repair, okay, the DNA repair. So P53 is activated in response to DNA damage. So if there is DNA damage, it can induce cell cycle arrest, which allows time for the DNA repair mechanism to go and fix that damage. And if the damage is irreparable, this P53 can trigger apoptosis. Can trigger apoptosis. Okay, apoptosis is what we can describe as programmed cell death. Okay, so there's no more point of fixing that part. Let's then kill, trigger this process of preventing the proliferation of this damaged cell. And so apoptosis is another process where p53 plays a key role, and so on and so forth. Many pathways that we can look into, for example, as well, angiogenesis. This is, the formation of the new blood cells. I referred to this in the first part of this module. So p53 can influence angiogenesis through its regulation of various factors like TSP1, ID4, and so on and so forth. So in general, when p53 is damaged, mutated, okay, we end up with disrupted set of pathways. This can lead to uncontrolled cell pathway, reduced apoptosis, increased potential for metastasis and angiogenesis, and so on and so forth. So certainly it is very important protein and gene in cancer biology, and it is an important and significant target for cancer research. Here's the list of oncogenes and the group of oncogenes as classified by the different classes and factors. Okay, different classes and categories. So we have oncogenes, list of genes that go into different groups, and based on that, there is a function related to that group. Something goes wrong, there is an associated specific type of cancer. So you can go through this list and see growth factors would relate to specific types of diseases. We are here at the transcription factor related oncogenes. Something goes wrong with this, we're going to talk about T-cell lymphomas and so on and so forth. Mutations in oncogenes can lead to the production of proteins that are constitutively active. This means that they are perpetually in an active state, regardless of the normal regulatory signals that would usually control their activity, which means they are in continuous activation. And this can have profound effects on cell behavior, again, leading to uncontrolled growth and division. So keep producing that protein. This is certainly going to be an issue. Let's look into identifying mixtures of cancer cells, and we've looked into this clonal and subclonal groups of cells, and we know now if we are doing sequencing here, let me ask this treasure hunt question. If we are interested in knowing the differences between the cells, what kind of next generation sequencing technique we can use, okay? And specifically what kind of RNA-seq method we can apply here. I think this should be straightforward, okay, to figure out. So that would be one solution to go and figure out, know at the granularity of the cell level and know the details at that level. What about just, the common mRNA-seq or RNA-seq that we are dealing with? Can it tell us about anything about the clonal or the clones? Like, let's take this an example through the IGV browser, and we have here a tumor sample with a matched normal one, and what we are looking here, what we are considering here is actually we're seeing some variants, okay? So you can see that we have some variants here, okay? Another variant here. So we have two variants. So how many clones are there in this case? Would require us to look more into these two variants, okay? In other words, are they part of the same, these two variants, are they part of the same sample and clone or not. Okay. This would require us to look at each single kind of read or strand. So if we zoom in a bit, okay, we do some calculations, we see that, okay, there are some changes. See, cytosine is becoming thymine and cytosine is becoming guanine in certain positions. Okay, that's what's happening here. And this always happens in cis, which means in the same strand. So I'm looking here at the same strand, right? You can see the same strands, we find the same changes. And if we look at the allele frequency in this case, we can see very similar numbers, right? This is around 58%, this is about 62%, very similar to each other in this case. And then we can look into this one, 38%, 42%, okay? And so this kind of association where we can confirm that they are happening at the same strands, these kind of variants, these two variants are happening, the changes are happening the same strands. In general, this confirms that they are from the same clone, okay? So we have the same clonal community. And what we're going to finish the lecture with is liquid biopsies, which is emerging and a new development technique. It's a minimally invasive medical procedure. It involves looking at the analyzing and looking at the blood, primarily blood, and it's an innovative technique. So the liquid biopsies are based on detection of circulating tumor cells, what are called circulating tumor cells or CTCs, okay, which is as the tumor goes between, especially when the tumor metastasizes, so it goes to other organ, some cells breaks in the blood vessels in the system, and so they go all around the place. So specifically in these cases, when fragments of tumor DNA also are released into the bloodstream from the primary tumors, they can provide crucial information. So this technique of liquid biopsies can detect cancer at an early stage. As any tumor cells or DNA can, if they're present in the blood, before a tumor is large enough to be detected by maybe other means. So it is useful. But again, you would be able to tell it can be limited because early detection of cancer using liquid biopsies can be challenging given the low concentration of tumor DNA in the early stages. There is still a chance that there is some release of these cancers to the blood system. Maybe it's localized, nothing is going on. And but again, there is some influence on the blood, then we can have a good detection of that. The good thing about this technique is that it's non-invasive, so you don't need to go and take a biopsy from the tissue. Okay. Now, one thing when there are, this is going to be maybe fast, early detection, and so on and so forth. Now, the golden standard is still a tissue biopsy, okay? But again, tissue biopsy could also influence the whole tumor. So when, I guess the protocol nowadays, when the cancer is spread in a metastatic phase or level, they may recommend to follow up with the progress and prognosis of the cancer to look at the liquid biopsy. Maybe this is, an easy way or accurate way in that case to see how much of development is there. Because once you have that detection, you are able to know lots of information. So we're done with this module. This module is here to tell us that bioinformatics plays a crucial role in cancer research, because cancer treatment nowadays is having genetic testing and genetic research as part of the protocol and as part of finding a treatment. As I mentioned earlier, although it's difficult, very difficult question, I guess, until now to answer. Is cancer a genetic disease or not, or it's not a genetic disease? As I mentioned, we don't see always mutations in the genes in the different cancer types. Some person can have a cancer, okay, not because they had mutations in any of the genes. So something goes wrong with the cell proliferation and cell growth and division. Yet the DNA of these cancer cells is corrupted and damaged, and there is some process responsible to block that, and it's not working in its best way. So really diving into this and understanding more and more about cancer is still very interesting, journey and curiosity journey to explore more and more. But we know that it's very important to do genetic testing. Even if the patient's having the disease, accessing the genetic information can help us, for example, next generation sequencing, to learn more about the progression and evolution and what type of processes or pathways are affected. And again, it's really patient dependent. We cannot really generalize. So conclusion here, next generation sequencing is very helpful and useful. It's something that we have to do and then see the results. It should be accepted as a standard. And then we look into all the information we can gain. And hopefully we are able to give advice. Like, for example, even like one of the things that I can tell you as a story to close up this with is, there was this patient that, where the physician made the recommendation, I recommend you move the breast and also remove both of the ovaries, okay? Because they are all, your type of cancer is hormone receptor, and so we want to kill the hormones in the body. However, what was not clear from this recommendation is removing the ovaries would bring an early menopause, which could have a lot of influence on the lifestyle of the patient, especially if they are at early stage. Think about a woman who's in the early 30s and so on and so forth. Now, when the person went to genetic testing, the genetic testing came with a list of all negative genes. And that helped, even though there were no mutations. The genetic testing informed that the decision that there's no need to remove the ovaries. This is not a genetic mutation or related, genes. We're not talking about maybe tumor suppressors or oncogenes. Everything seems in general normal. Maybe something we couldn't spot. Again, how much these tests are comprehensive, this is another thing. Next generation sequencing can give us a wide spectrum of things that we can look at, while these genetic testing happening now are focusing on a specific set of genes and gene panels. But anyway, that informed you don't need to take that surgery and remove the ovaries. So that changed the life of the person based on their genetic testing. They are happy now. They will not go through early menopause. Of course, they need to have the treatment to suppress the ovaries, and which may bring some also side effects, but at least they're going to stay there. They can have in the future a normal lifestyle. With that, I hope you will be safe from any cancer, or you will love people, all the people we know. It is something we are struggling with in this era. So may this be the last module, which is where we, give a recording. I might give something for the, in the final presentations. Let's see, or maybe, in an office hour. But I hope you have enjoyed this course, and then we are going to follow up with each other. Coming few weeks are very critical for working on the project. Okay, so good luck, guys.